So, welcome everyone. This is week 6 of the course, moving to module 6 about alignment and reference genomes. So, we will go and look into a few concepts related to alignment, specifically RNA-seq alignment, and dive a little bit there and discuss some of the steps that are needed to achieve that alignment. This is kind of going to establish thinking of what algorithms or what kind of algorithms are used. And now you see, I mentioned algorithms. You understand that we moved from molecular understanding, molecular concepts in the course. Next, the biotechnology side and the technologies and the machines and the experiments that would be established in some setup in the laboratories to get some samples, prepare them, give them to the machines, and then get the output reads. Now it is the time where we need some programs. So this is the journey of a bioinformatician um, who needs to understand something about the technology aspect, and also about algorithms and programs that are needed to analyze the data. So I hope so far, you're doing very well in the course. Um, Let me remind you, it's um, uh, time to reflect on your own projects. This is already half of the semester, let's say roughly passed. So I guess all teams had their papers selected, uh, provided the feedback. So what I'm hoping for is you start reading. You have one paper to work with now. Make sure you understand it as a full team. My advice would be, everyone read it, um, and then have one meeting where all come together and discuss this paper. Make sure everyone has done their part in reading the paper. Um, I wouldn't personally think that's the way to go, like to say, well, at this stage, take this part and read it. I will take that part and read it. I don't think that is the way to go. Everyone should read fully the paper, understand what's in there, and then, uh, you know, it might be the case that when you read the paper that lots of terms or concepts or ideas are not clear, so you spend time decoding those. Okay. um, so maybe a couple of reads would be needed, so give yourself the time, and then the whole team members, when they come and discuss together, they make sure that they are all on the same page. Next is defining a plan. Okay, so I think what would be also reasonable is to say, well, you know what? Based on our discussion as a team, what about focusing on this section of the results? Break it down. Start with something and say, okay, this seems an interesting figure in this paper. This seems an interesting table. This seems an interesting number. Let's see if we can reproduce that. And from there on, you're gonna start your journey, right, by looking into data and start to go through all the challenges. Um, I would say generally everyone stick to your paper from now on. Don't go back and say, "Oh, I've discovered this. I want to change it." Take the challenge, and everyone, I believe, may find some, kind of questions, some kind of, I don't want to say surprises. Hopefully, you don't see any surprises, but this might happen, which is a case where you may find there are no sufficient information. So I hope everyone is planning their projects, the teams. This is going to give you some time. Another thing that I want to do as part of the check-in here is going on the Discovery server. So last week I had this online tutorial. So I hope that you have followed the steps. This week we'll have another exercise. From now on, generally expect some, I believe about on a weekly basis, we will have some exercise on demand. So make sure you have got into the server. Some students, I received requests to, that they want to join the server, which is part of Module D, which was, right at the beginning, even before starting the course, it was released. So anyway, make sure you don't delay this anymore. So make sure you access the server. And the final thing, it is documented on my own side who has been watching the lectures. And so I would still invite everyone to watch the lectures, the recordings. And this is going to give you bonus 2% at the end if you keep answering and addressing the the treasure hunt questions. And also it brings my attention to your activity level, which will make my evaluation positive. This is inside the course where I'm happy to collaborate, discuss anything that would be of matter related to grading. But also after the class, I don't mind to give a reference letter for the good students. So please watch the lectures, and those who have been watching it and sending me the answers, thank you. I am so happy. This is one of the things that makes me very happy in the course, to see that you are watching and addressing the questions. So as we go through this module, and again, we'll see that it's sort of another shift. So after we looked before into the technology aspect, now we'll see, it's more diving into bioinformatics, maybe the software algorithms side of that, okay? So today we will explore a little bit of that. Again, it's not going to be comprehensive, but we will do our best to highlight specific concepts. Some of these concepts, might be new for you, so we'll try to, again, break it down. And please, if you have any questions, share them with me. If you have any things that you're interested to learn about, also share those with me. I will try. I got some interesting suggestion about quantum computing, for example, how we can use it. I'm still looking for a place where we can fit that in. I'm not sure if I will do, but again, I think it is very interesting. So share some thoughts with me similar to that anytime. The floor is open. So more about algorithms, and algorithms would mean steps that we use to solve a problem given an input, specific input, and that's going to give a specific output. So when we want to develop an algorithm, we are looking at the problem definition, meaning what kind of challenge we are working with. And the communication would be between both sides, meaning the one who is proposing the problem. Let's say, for example, the biologist or the chemist, they need some help because they have high throughput data. They need some algorithms to solve the problem. And from the other side, I'm going to say we have the bioinformatician who has the programming skills. So both they communicate and try to establish, good communication to understand what's going to be the input. Knowing the input and is very important to know, and the goal as well. So you give me the input and you tell me, for example, you say this is the genome text. So what is the goal? So it is different thing when you say, well, I'm looking for origin of replication in that text. I'm looking for transcription factors in that text, okay? And I'm looking to do maybe alignment using that text. So these different goals would guide me to different way of thinking. And so we're going to dive more into the problem specifications. So understanding the problem is very critical in this domain, guys. And always knowing more biology and more computer science related concepts is going to bridge that gap to develop a better solution. So hopefully through this lecture, we get to bridge some gap when it comes to RNA-seq alignment. And in general, when we're talking about, RNA-seq alignment, we're referring to sequence alignment. And this is a method used in bioinformatics to arrange biological sequences. So you may have some reads that are DNA, RNA, or protein sequences, and you want to identify regions of similarities and start mapping those toward the goal of building functional or structural relationship. And so through this process of alignment, for example, RNA-seq alignment, and after arranging the sequences to identify these regions of similarities, we can achieve several goals. For example, we might be able to align a newly sequenced RNA to the database of known RNAs. This is helpful in predicting the function. So when you have a novel or a new sequence and you go to your database of documented RNAs, and so this new sequence is going to be mapped into those ones, aligned to some of those, is going to help in figuring out the function. And this is part of what is called functional analysis. In medical and genetic research, aligning RNA sequences from different individuals can help, for example, in identifying some of these variants associated to specific disease. So think about genetic testing, for example. If I want to do genetic testing for a breast cancer, which is now part of, this is a very common disease nowadays, which is unfortunate for many people, and part of the standard protocol of treatment now for breast cancers, and I hope everyone recovered from this and survive it, is genetic testing. So now part of the treatment protocol, okay, we're going to start chemotherapy. We may have radiation therapy, hormone therapy, all of these types of therapies to comprehensively and try to tackle this serious cell division issue in cancer. Genetic testing is going to be part of these tests. So genetic testing, to be done, they're going to screen the patient for specific genes. So they should have a library with a list of mutations that identify a reference if you have this disease or not. So your sequence information is needed to try to align those and see what is the chance if have that mutation, and so you will get recommendations for the treatment, okay, and so on and so forth based on that. So studying alignment or reference genomes is certainly crucial for many reasons. Gene discovery and annotation. So again, we are aligning sequences to a reference genome, which helps in identifying genes, regulatory elements, and other functional elements. What about variation detection? Okay, we talked about variant detection. So we talked about some types of variants before, and I mean genetic variants, like single nucleotide variants or single nucleotide polymorphisms, insertions and deletions, indels, right, in the previous lecture and previous module. So these variants can be associated with phenotypic traits or diseases as part of sequences. Think about also personalized medicine. So again, as genomic medicine advances, knowing the patient's genome and mapping it to a reference can identify some genetic risk factors. So I personally think this area in the future would be very well developed. Now, again, we don't know to which extent personalized medicine is going to work for all diseases. It's going to depend on many factors. But I personally think it's going to be, I don't think it's a me. You need to be futuristic to come up with these conclusions. What did you saw? The big drop in the technology expenses, novel technologies develops that are of USB size, benign nanopores that we have discussed before. So imagine the future is going to be very accessible to everyone to know their genomes. Okay, and based on that, hopefully we can give early diagnosis of what may happen in the future, and then some recommendations, and so on and so forth. So alignment is certainly very important. So let's dive into specifically RNA-seq. This is the next generation sequencing. One might ask why RNA-seq? Well, this is the most widely used technology nowadays for sequencing. So knowing alignment of this would certainly be helpful for all of us in this class. So what do we see here? Okay, this this may seem a bit colorful for some. Maybe some of you know some terms here, and it may seem like a map for the train in the city. We are trying to go from metro from one point to another point, and so on and so forth. What do you see here is a pipeline, okay? A sketch of what we may call a pipeline, okay? So what is a pipeline, right? We have pipes to flow the water at home, right? And the pipe means basically I have one step that takes the input from the previous step, and then we keep going. So I move one step, I execute a program, take input, give it an output. The next step, I take the output of that program, give it as an input for the second program, and then I move on and repeat. I take the output of the second program, give it as an input for the third program. And so this is what we see here. Okay, the output is going to feed this as an input, it's going to feed this as an input, the output is going to feed this as an input, and we keep going. And in general, this is categorized into 4 areas. And this is a very nice summary of what we would call as an RNA-seq, alignment pipeline. So you have 4 or 5 areas, right? So pre-processing, this is the area of pre-processing. So once you have FASTQ files, what can you do with those? Always, it's a standard way of thinking, okay? We don't take data for granted. Today we discussed some issues of bias, and we may have certain errors or so. So why we don't apply some filtering, or why we don't use some tools to visualize the quality or examine the quality in these files, and then based on that, take some decision to do some trimming, some filtering, taking out some reads, some adapter sequences, things that are not necessary, okay? And bad reads, for example, correct maybe for sequence errors, and so on and so forth. So this would be part of this first stage, that, you give me FASTQ files, you have executed the experiment, okay, that is fine. With all due respect, we still need to do our side as a bioinformaticians from a software perspective. We want to systematically do an analysis and see the quality of data. It could be the case that the data is not of high quality for some reason or another. So we bring this into attention of the experimental side. Now, stage 2 here, okay, it's genome alignment and quantification. Genome alignment and quantification means I need to take the reads I get from the FASTQ files and another input, which is the genome. So I'm going to show you an animation shortly to illustrate this point. So we need to map the reads to the genome and start quantifying, doing some calculations, like count the number of times we have that specific transcript being matched to a number of reads, and we use that number to indicate eventually gene expression. Okay, so there are many tools to do that. What you see here is there's star splice transcript alignment to a reference. This is an alignment tool specifically designed for RNA-seq data. It is known for speed and accuracy, and especially when mapping across splice junctions. Okay, there are these kind of what you may think of as gaps. In fact, in this module, part of the activities section that we'll have in this module, we're going to work with star software, and we'll try to run it on the Discovery. Okay. HiSat, there are developed of these. There are, as I said, set of tools now. HiSat uses a different alignment strategy than star, and it is a successor of what's called Top Hat 2, and it's also designed for aligning RNA-seq. So there has been development of very interesting. I'm not going to say just algorithms; those are tools, including algorithms. In other words, you can download these tools, and they are well documented, and you can run them from the command line. Sometimes you have online interfaces to give you options, like if when I talk about a Galaxy server, for example, and we are able to execute these programs. Now, what they need, again, they need a genome reference file. Other tools that are pseudo alignment. In this case, some of these tools is what's called Salmon, and there's Callisto. Those, they don't need the reference genome. They are referred to as they're referred to as alignment-free methods. So instead of aligning reads to a reference genome, these tools like Salmon reads to a transcriptome. Okay, so we can have access to genome file. So usually somebody has established that genome file for us. So you can go to some databases. We'll see this in the coming module, how some databases offer us an access for genome files. So we can use them with these tools. Someone will come and say, okay, where I better get the genome file? Well, if it is one of the species as known, already characterized, the information is there, you go and download it. If not, you do what's called de novo assembly. So you come up with your own kind of reference genome, and then you use, kind of the ortholog analysis, try to derive the functions and so on and so forth, and build your own genome. Your own annotation is a bit, demanding process. But anyway, generally for many of the species, we can download the genome file and the transcriptome file. And of course, there are differences, right? Genome file would have exons, introns, would have things that information that would relate to the chromosomes and so on and so forth. Transcriptome, we're jumping directly to the transcripts. So there would be differences, right, in terms of what is a transcript and what is a DNA sequence and so on and so forth. So this is stage, well, 2 or 3. You're going to choose either 2 or 3. Is interesting. 3 usually may seem to be a faster option. Accuracy is, the papers there claim that they still achieve good accuracy as compared to these ones. So anyway, depending on the nature of the application, and if you have the reference genome, the transcriptome, what is the quality of the transcriptome, and so on and so forth. Some of the questions that one may need to ask and resources you have before you decide on maybe having one of those, or maybe a few of those, and then systematically compare the output. Then we move to post-processing. Now, one of the things that can happen here, okay, yes. So when we go to the this stage of post-processing, like using what's called SAM tools. Now, this is a suite of programs for interacting with the high throughput sequencing data. So usually the output from the previous step is what's called a SAM file, and then we want to change this to another format of file, which is binary alignment file. So we are going from sequence alignment map, okay, to the binary alignment map. Why? Because this is, if you go to the binary, always you're looking into a smaller size of file. So these steps, one would say, why do we need to do this? Now, in terms of alignment and these kinds of tasks, you need to understand when we look at these softwares, like I've had this experience personally. You end up for one experiment having maybe, let's say, as we said before, one treatment, one low dose, one medium dose, one high dose with few replicates each, let's say 4 or 5. I end up with 4 times 5 replicates, or for 16 to 20 samples. So let's say roughly 20 samples. Each is going to have 2 FASTQ files, one forward right and one reverse, something we highlighted in the previous module as well. So we'll have 2 files, each roughly is about 2 gigabytes. And so 4 gigabytes for one sample, and 20 sample end up with 80 gigabytes. And that is only one experiment. So when I've worked on one project, we didn't have one experiment. We had maybe, I don't remember, like maybe you can think of 60 experiments, 70 experiments, okay? And each one would be roughly, as I said, 80 to 100 gigabytes of data. So this is going to end up with terabytes of data. So you need then to understand, it is one of the things that we always need to think, storage. Storage is going to come as a challenge for us in this domain. So having sufficient resources, backup, where do we put our files, or how do we build the backup protocol for our lab or company or research lab or government lab, and so on and so forth. And also how we execute the steps in the middle, trying to make some compression. Like in this case, going to the BAM files, and also running some other possible post-processing tasks. Now, finally, this is final quality check. Let's do a quality check again and make sure everything is fine, and then run some downstream analysis along the way, like doing differential expression analysis, and see if the results make sense, right? And usually here is where we start doing more and more visualizations, okay? And I personally love visualizations in this domain. Think about the heat map, for example, something hopefully we'll see later on. So yes, I spent a little bit time with this figure here, because we're going to dive more into concepts as compared to the technical aspect. So use this as a reference for you if you're interested to, in the future, to know what kind of tools you can run. And know that many of these here are just the name of programs, like FastQC is a program, TrimGalore is a program, FastP is a program, and so on and so forth. Star, HiSat, Salmon, DSIC2, and many of those SAM tools, they are all programs. And so you tell me, oh, to execute RNA-seq alignment, I'm going to end up with 20 programs or 10 programs or 5 programs, 3 programs. Usually it's not that huge. It's your own decision. Usually you use maybe 4 or 5 programs, and then you build a pipeline. Okay, you build a pipeline. Good. So my first treasure hunt question for you is, what did you find interesting here? Okay. What did you find interesting in this part of the story of this pipeline? If nothing, just say nothing, and it's an accepted answer, because this is just for, some participation. Okay, as part of the alignment process, guys, indexing, okay, we have indexing is one of the necessary steps, okay? What is indexing? Okay, we started to jump to some terms, okay? Now, let me remind you that, well, there have been some samples that were prepared that went to the sequencing machine. We got the reads. Now we have this This is always where, once they are at this stage, they're going to ask you as a bioinformatician, please jump in, execute, and do your own part. So in this case, what is your own part? You want to get the read mapping and then generate the final results, which usually typically looks as a table. I want to remind you, before I've showed you a tool which is called Network Analyst for some, and gene expression related analysis, downstream analysis. So I would certainly suggest you go see that. There are some examples, and you would see some tables that would help us to see genomic variants, gene expressions, and what kinds of information relates to this disease, and control groups, and so on and so forth. So as part of the read mapping, there are some stages, okay? There's what's called indexing, pre-alignment, filtering, and sequence alignment. So we're kind of when I understand what happens in this phase of read mapping, this is sort of, diving more into this stage, okay, stage 2. This is sort of diving specifically into this, because this is really a very important step, and it's a stage and the focus of this lecture. So indexing, in this case that we're looking at, is mainly before aligning short sequence reads to a reference genome. So here we have a reference genome and we have some short sequence reads. Usually before doing that, the genome itself is indexed. And we're going to dive into this. We'll see how to build some of these indices. We're going to go into indexing in good in depth. The idea is you have reference genome, you have one file of FASTQ, you take the reads. If you have to go sequentially through the reference genome, meaning read it fully for every time you want to do some comparison, let's say that this is, imagine for the moment that this is just the genome text, okay? And then all we want to do is, again, and let me do this, okay? And so all we want to do is we want to see how many times AC appears here. Here's the question. How many times ACC appears here? So this is another treasure hunt question. Please share it with me. I'm going to give you a moment. And so what you're going to do, probably you will take this and then map it. Okay, this is ACC. Okay. Now, next mapping is to shift this one position. So here we have ACC coming at this position. So there, if I compare this to this, there is no perfect matching. This is C, this is A. So I'm going to jump this over and shift it another position. Okay, no perfect matching. It's here. This with this, they are the same, so I end up with another count. So I increment, so now I have 2 counts, and I keep going. Okay, this might be an easy text to find, so I keep going. So what I have done is I took one read. I want to know if it is in my genome, and find the location of that, because I want to do further things. So once I align it here, I'm going to store these positions here, okay? And then I'm going to keep going to construct my full information, and use that to do the mapping, the needed mapping. So, and I'm also counting as I go through this, okay? So anyway, while we are doing this, if this is going to be the process, like each time, for each single read, go through the whole genome text, which is for the human is 3.2, about 3.2 billion nucleotides, roughly 3.2 gigabyte text file. And imagine for every single read, you have to do this whole process again and again and again for every single read. Now, we have millions of reads, if not billions, right, in these FASTQ files. So we have millions and billions sometimes. So when we take these reads, and if we go for each one, you have to go and fully read the 3.2 gigabyte file. It's not efficient. Now, some of you may come and say, oh, how can we do it differently? Okay, so this is what we're going to look into today's lecture. So anyway, that is indexing the reference genome. And so eventually it's going to help us to reduce the number of, to reduce the number of seeds. So seeding is strategy where shorter fragments that, or call them seeds, from the sequencing reads are used to find potential alignment locations to the reference genome, potential alignment locations. So these are the fragments that we try to map. So by selecting and reducing the number of seeds, we can optimize the balance between alignment between alignment speed and accuracy. Now, another thing is that as we are trying to understand this, we take these fragments from the sequence reads, and also we're trying to map to the genome. So once we break this whole thing down, instead of comparing again everything to everything, we're trying to control the process. So and that's why we are eventually going to reduce also data movement as we use efficient data structures. Another stage, okay, now we have our reads, right? We have our genome index. So we did not start the alignment yet, okay? But we have built some information, some genome information that we can use. And then, we have done some organization of how we can access this reference genome. Next, we're going to go to the pre-alignment filtering. Pre-alignment filtering is a necessary step because it quickly eliminates many unlikely alignment locations. So, for example, there's what's called Q-gram filtering, which is the Q-gram refers to a substring of length Q, okay, from the original sequence. So if we are able to count the Q-grams in both the reads and the reference genome, these substrings, areas of potential similarity can be identified, bringing the focus on allowing for focused alignment efforts. So in this case, well, we might not, think about this. The way this is going to happen is all of these principles, pigeonhole principle, and these counts, and so on and so forth. The principle here is, you know what? You have reads. Let's say I have 1 million reads, and you have a genome. Okay, let's say it has 1 billion areas of interest. So, do I need to? Again, we understand we don't need to compare everything to everything, and we know that we're going to have a strategy to access the genome locations. But again, are we really interested in everything? Why we don't just, we know that this is our area of interest, so why we don't filter? And this filtering can, something we subject the reference subsequences to. And so when we have this data structure built, we can decide to remove some of its part, and then that's going to help us in reducing the number of comparison. Eventually, we're going to do sequence alignment, where we try to align the different sequences using different methods or classes of algorithms. Sometimes what's called dynamic programming is used, for example, like what's called Smith-Waterman, Smith-Waterman, or heuristic methods, like BLAST technique, seed and extend methods like Bowtie, and so on and so forth. So there are many softwares, many solutions into this class. So back to refresh with central dogma of biology. So we had DNA replication, and then we get the transcription through the help of RNA polymers to get the RNA sequence, and then you getting the help from ribosome that will use transfer RNA units to start building the amino acids, and so we'll get the protein, and so on and so forth. So through this stage, we're going to get eventually, if we're preparing a library with this information, we are having sequences. We know now, well, at this stage, we understand whatever is generated in terms of RNA-seq. So in terms of RNA-seq information, like in this case, think about having messenger RNA, okay, that would eventually relate to a protein. Now, this RNA here, if I have eventually some reads that represent this part, and we want to map it to, the full genome text and know which area of interest is that, we need to keep some things in mind, okay, some things in mind, okay? For example, when we discuss the DNA and the genome text, we say there are areas that are, we refer to exons and introns. And if I would like to remind you that exons are coding, okay? And so, and they contain information for making a protein. So the RNA in this case, okay, now if we look into just want to illustrate one idea. So imagine this is the DNA text, okay? So we have some areas that we refer to as exons, okay? So there is this one, okay? And in between, we'll have introns, okay? Introns, okay? And now, after, the process of translation, and there's this step of splicing, we get mRNA text, okay? But we get the mRNA text by removing these introns, okay? So end up getting eventually, and we have discussed this before, so we may end up also with different exon arrangements. So this could jump here, and that could go here. Anyway, so we get some, let me use a different color to indicate the mRNA part here. Okay. Okay. Now remember, this is what I'm using the lab to generate my reads, right? So if I want to take those, map it to the original reference genome for alignment, remember that this is not a direct mapping, because we have to figure out where we had these gaps, right? Entrance, and if this read is going to map in that way, overlap here or there, and so on and so forth. These are some of the, I would say, tricky questions, or or questions that one need to deal with when they are coming up with their own algorithm. They need to understand the details into how to do the full alignment process. So with that, we move on, and we understand we're going to have some species as part of our experiment. We're going to extract some sample, do the execute our kit in the lab, and then extract the DNA, for example, or the mRNA, right? And go and, of course, subject this into certain technology, which is going to make fragments. We understand next generation sequencing, like Illumina sequences, and so on and so forth. This is how they work with short reads. And what do we get? We get these short reads to our program. Eventually, this is a set of short reads that we can see in a FASTQ file. Let me remind you what we had discussed last week in the previous module. And also, let me remind you that with the in-class activity that we had last week, where you would have been able to see actual reads for true data and play with that. So anyway, we have these reads. Are we done? Nope. We need to understand. If we don't have a way, well, let me put it differently. We are solving the puzzle, right? Let me remind you back about something. You remember when we had the newspaper example, and then boom, pieces everywhere from repeated cases, or the shotgun sequence, and so forth. So now what we want to do here is to solve a puzzle, and solving the puzzle with no reference information is sophisticated. So since now we have a way to do it, which is a reference genome, meaning somebody already made an effort to establish the genome. And this was, as you mentioned before, this was the Human Genome Project, was actually to build a reference genome. Okay, and we will look into this today. We'll see summary of some information related to reference genomes. Okay, reference. This is the reference genome. So the reference genome is already constructed for us, and it's there. Now I have my own reads, okay, and from my own experiment. So I can use this to do the alignment. Okay, so in this case, oh, this text aligns here, this one aligns here, and this one aligns here, and so on and so forth. Right? So eventually we get to see some quality scores once we are done with this part. For example, one of the quality scores here that we're looking at is what's called read coverage. And read coverage refers to the proportion of a genome or transcriptome. It could be genome or transcriptome that has been sequenced. Okay. Okay. So for instance, if 90% of a genome has at least one sequencing read mapped to it, the read coverage is 95%. Okay. So here's how we can build that. Read coverage here equals the number of bases mapped. So this is, if I try to check here the mapping, this is mapped 1, 2, 3, 4, 5, 6, keep going, extended 7, 8, 9, 10, okay, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Okay, so number of bases mapped is 22. And the length of this reference genome is 25. So the read coverage here is 88%. And this gives me an idea about the completeness assessment. It provides an estimate of how much of the target genome or transcriptome has been captured in the sequence experiment. And also it helps me to identify what's called a gap. Okay, for example, there might be areas with zero or low coverage. Okay, maybe in this case, I don't know if this is here. Imagine this is, yeah, this seems a gap here. Okay, so there might be areas with low coverage or zero coverage, referring to areas that are difficult to sequence. Okay, this can guide further experiments or highlights areas of additional checkup and so on and so forth. So this is one measure that is widely used, read coverage and next generation sequencing. It's one of the things you want to look at, how much coverage was achieved with the given technology that is used. Another concept is the read depth, which is just the number of reads at a given position. And read depth refers to the number of times a particular nucleotide position, in this case, okay, each single position is read during the sequencing process. So if a specific base in the genome is covered by 30 individual sequences, for example, the read depth is at that position, at that position, okay? The read depth at that position, okay, is 30x, 30 times, okay? And this is again helpful in knowing some quality information, building some quality control. For example, the read depths would help in establishing confidence about variants, because higher read depth allows for more confident variant calling. And if a variant is observed in a significant proportion of reads at the high depth, it is likely a real variant as compared to just a sequencing error, okay? So these sort of questions or ideas that we can use and so on and so forth to know if this is, more of an error, more of some interesting insight that we have. Okay, so I'm gonna leave this to the second part.